This lecture covers nucleic acids. So nucleic acids are the fourth macromolecule that we're going to look at. We've covered carbohydrates, lipids, proteins before this. There are two types of nucleic acids, DNA and RNA. And the monomers of nucleic acids are called nucleotides. So nucleic acids, like carbohydrates and like proteins, they are also polymers, and so they are made out of small building blocks, monomers, and they are specifically called nucleotides in nucleic acids. The function of DNA is to be the genetic material. The function of RNA is to help with protein synthesis and to actually make copies of the genetic material. We'll look at this more at the end of the semester in chapters 13 and chapter 14. Now let's talk about nucleotide structure. What are the components of a nucleotide? There's actually three of them. The first one we've talked about before as a functional group, and it's called phosphate. So it has a phosphorus atom surrounded by bonded to, 4 oxygen atoms. The second component is a sugar, specifically a pentose sugar or a 5-carbon sugar. Now, if we're talking about DNA nucleotide, then the sugar is called deoxyribose. If we're talking about RNA, then the sugar is called ribose. And on the next slide, we'll see some differences in the structure. But either way, the sugar tells you which kind of molecule you're talking about, because DNA actually stands for deoxyribonucleic acid. RNA, the R stands for ribose, or ribonucleic acid. The third component is a nitrogenous base. And you can pick from either adenine, cytosine, guanine, or thymine if you're talking about DNA. If you're talking about RNA, you can pick from adenine, cytosine, guanine, or uracil. So uracil is only found in RNA and thymine is only found in DNA. So these are the three components. Here's pictures of just the bases by themselves. We group the bases into two groups called pyrimidines and purines. And we can see that cytosine, thymine, and uracil are all classified as pyrimidines. One of the things we can notice when we look at the structure of pyrimidines is that they all have a single ring. Below we have the purines, and that is adenine and guanine. And if you look at the structure, you can see that they actually have 2 rings. So structure-wise, they differ in the types and the number of atoms, but they also differ in having a single ring versus having 2 rings. When we look at the structure of DNA on the next slide, we'll see why that's important. So how do you remember if what you're looking at is a pyrimidine or if it's a purine? So look at how pyrimidine is spelled. It has a Y. Look at cytosine. Look at thymine. They also have Ys, so they're pyrimidines. And you can just remember uracil fits because it substitutes in for thymine. If you look at purines, notice that guanine has a U. Adenine doesn't have a U. You're just going to have to remember that one. But if you remember the other ones, if you remember who's a pyrimidine, then you can figure out who a purine is. So this is what a total nucleotide looks like, a specific one. This is, again, the cytosine nucleotide, and it has a phosphate group. It has a deoxyribose sugar, and then it has the cytosine nitrogenous base. Let's talk about DNA structure overall, the whole molecule. It's called a double helix. And we're going to talk about in chapter 13 later this semester about how we figured out that it is a double helix. It's often referred to as either a spiral staircase structure also, or a twisted ladder shape. So the handrail or the sides or the backbone of the molecule is repeating phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. And that backbone and the bonds between the sugars and phosphates is called a phosphodiester bond. And it's actually between the oxygen of the phosphate group and a carbon on the deoxyribose sugar, or a ribose sugar if you're looking at RNA. So that's what holds the two sides together, the two strands together along from top to bottom. Now, because it's a twisted staircase kind of shape, it has steps, and those steps are actually made out of base pairs. And the two base pairs, or all the base pairs I should say, are held together by hydrogen bonds between two bases. And in DNA, adenine always bonds to thymine, and guanine always bonds with cytosine. And if you look at the picture, you can see the top base here. On the left is thymine, on the right is adenine. Thymine is a pyrimidine, remember the Y. Adenine is a purine. So we always have one pyrimidine to one purine. And if you look also, thymine has one ring, adenine has two. And the little pink dots represent two hydrogen bonds between those two bases. If you look at the base pair below that, it's a GC base pair. On the left-hand side, we have guanine, which is a purine, bonded to cytosine, which is a pyrimidine. And again, notice we have a two ring bonded to a single ring. And this base pair actually makes three hydrogen bonds. You can see three little lines there with the pink dots. And so this is the way it works. If you had a wrong base pair, let's say maybe G bonded to G, then it would stick out. It would be too wide. Having one ring and a double ring always as your pairs helps keep the diameter of the molecule nice and straight. The other interesting characteristic of DNA structure is the fact that the two strands are anti-parallel, which means they run in opposite directions. So let's look and see how we can see that a little bit better. If we look at just the sugar here, the deoxyribose sugar on the left-hand side, notice that the oxygen atom is at the top, at the little peak. If we look at the sugar on the right-hand side, notice that it's upside down. That oxygen, the little peak, the top of the sugar is facing down. And you can see that along the strand. So this is, I think, the best way to visualize what we mean when we say it's anti-parallel and that they run in opposite directions. Again, this is going to be important to us in Chapter 13 when we talk about how DNA can replicate itself. Let's compare and contrast DNA and RNA. One thing that's different is that DNA has deoxyribose, while RNA has ribose. Now they both have sugars, but they're slightly different. The arrow on the right-hand side shows you where the ribose has an oxygen, a hydroxyl group, where deoxyribose just has a hydrogen. Another difference is a base. Remember that DNA has thymine, RNA has uracil. And if you look at these structures, you'll notice they're almost identical, except for thymine has a little extra carbon on the side. And this is why when DNA and RNA interact with each other, adenine can bond to uracil, and adenine can also bond to thymine. We'll talk about that a little bit later, again in Chapter 14, when we talk about protein synthesis. The overall structure of DNA is double-stranded. It's a double helix. RNA, however, most of the time does its work as a single-stranded molecule. Again, in Chapter 14, we'll talk about the different types of RNA that are involved in protein synthesis, and we'll see that they're all single-stranded. The function of DNA is to be the genetic material. It contains genes. Genes are instructions or recipes that contain a code that need to be used to make all the proteins that run organisms. RNA's job is to copy that genetic information, to copy the recipe, and help with protein synthesis, with actual assembly and making of proteins. Again, Chapter 14, we'll get into the nitty-gritty, dirty details. And lastly, DNA is located in the nucleus. It's in chromosomes, and that's where it hangs out. It needs to be protected because, again, it has all the instructions on how to run the cell. So we need to keep it locked up and safe inside the nucleus. RNA, on the other hand, can travel between the nucleus and the cytoplasm. It's going to copy the information from the DNA and bring it out to the cytoplasm, where it's going to be assembled at a particular structure called a ribosome. Again, Chapter 14, we'll get into the dirty details. And that's it for now.